Sarah Ashley, you can start.
Hi everyone, welcome and thank you for joining us today. Sorry, I thought I was muted on. Thank you for joining us today for the NCI Obesity and Cancer webinar series, Microbiome and Cancer Consideration. My name
Your volume's a little low.
I'll speak louder. My name is Sarah Ashley and I will be your WebEx technical support host. Before we dive into today's meeting, I wanted to provide some quick information on the webinar platform. All lines have been muted upon entry and will remain muted for the duration of the webinar. Submit questions throughout the presentation, chat, Q&A box, and select all panelists. Moderator will ask your comments and/or questions on your behalf at the end. If you require technical assistance, please feel free to contact me, the host, and I'd be happy to assist you. Captioning is available and a link has been provided in the chat box. I also wanted to make you aware today's webinar is being recorded. And now I'd like to turn it over to Phil Castro.
Phil?
The volume was very low on that. I'm not sure everybody heard anything, but in any case, welcome everyone on behalf of the NCI Obesity Working Group's webinar committee. I'm very pleased to introduce today's webinar topic entitled Obesity, the Microbiome and Cancer, Mechanistic Considerations. We have a really outstanding group of speakers today to present this topic and to discuss some of the basic biology that links obesity, the microbiome, and cancer. The presentations today will explore some of the pathways, models, and bacteria that are involved in influencing obesity and cancer outcomes in 3 types of cancer: colon, breast, and liver. But before we hear about that exciting work from those specific examples, it's my great pleasure to introduce Dr. Stephen Hursting from the University of North Carolina. Steve is an alum of the NCI Cancer Prevention Fellowship Program, and so he has deep roots at NCI. He is currently the director of the UNC Nutrition Research Institute and distinguished professor at the Gillings School of Global Public Health, as well as a professor at the Leinenberger Comprehensive Cancer Center. And Steve's been assigned the rather difficult task today of giving a brief introduction and overview talk on how microbiome alterations may be affecting cancer outcomes in high-risk obese populations, which will nicely set the stage for the talks that will follow. And so with that, I'd like to ask Steve to come up and start us off.
Thank you, Phil, for the kind introduction, for your leadership and advocacy for microbiome and cancer research at the NCI the past 2 decades or more, and for organizing this terrific webinar today on the role of microbiome in the obesity cancer link. It's my honor to lead things off, and we'll first talk about the current evidence on the impact of obesity on cancer, and then we'll raise a few key questions that our distinguished panelists will address regarding this complex relationship between obesity, microbiota, and cancer. So let's start with okay, I'm not advancing for some reason. There we go. Let's start with the rigorous expert report series from the American Institute for Cancer Research, World Cancer Research Fund. I've had the honor of serving on this initiative for the past 24 years. I joined right after the initial 1997 report. And one thing that's really a striking aspect of this initiative is how strong the obesity cancer connection really sort of evolved as the data got better and better over the years. And so the latest report showed convincing evidence of 12 obesity-associated cancer types. I was also part of the IARC, the International Agency for Research on Cancer, working group on body fatness and cancer. This emerged into their handbook number 16 that was published in 2018. And this took a very different approach. Instead of the systematic review meta-analytical approach that the AICR initiative did, this looked at the obesity cancer connection through the lens of the IARC carcinogenesis protocol. So it was really an interesting exercise. And so despite the very different approach, strong concordance between the two initiatives here, where this group, there was an epidemiologic team that found 13 distinct cancer types. So they added meningioma to the picture, but very, very similar findings despite the different approach. The other challenge that this team was asked to address was whether intentional weight loss in people with obesity reduces the risk of some cancers. And the answer is a qualified yes, based primarily on bariatric surgery studies. But the number and quality of weight loss studies was deemed insufficient for formal evaluation. So here's a major gap in the field. Yeah, we know obesity is a strong risk factor and progression factor. What do we do about it? How do we reverse that is a key unanswered question at this time. I was part of an animal models team, and we found that obesity consistently promotes cancer in rodent models of the same cancer type shown to be associated in humans. Now, the number of studies for many sites were limited, but really, again, a clear picture similar to the human. We also addressed the opposite end of the spectrum, calorie restriction. There was a large literature in animal models regarding the anti-cancer effects of preventing adult weight gain with calorie restriction. And again, there was sufficient evidence that that was a strong preventive effect in the literature. Once again, we were challenged with looking at the question of whether intentional weight loss can reverse the pro-cancer effects. And once again, this just hasn't been studied to a great extent yet. And again, a key gap in the field. I was also part of a mechanisms team. So there were three groups, the epi, the animal side, and the mechanisms team. Cornelia Ulrich and colleagues were part of that with me. And we found a number of established and emerging mechanisms that underlie the obesity cancer link. So once again, using the IARC mechanism, weight of the evidence on mechanisms criteria, we found largely the literature has been focused on systemic growth factors and inflammatory factors and their relationship to cancer risk and progression. So increased insulin-like growth factor 1, the increased leptin-adiponectin ratio, number of cytokines, chemokines, and vascular-related factors have been well studied, and there are strong links here. So these were established mechanisms, particularly through receptor tyrosine kinase signaling, particularly through the PI3 kinase AKT mechanistic target of rapamycin pathway. A clear picture that obesity drives signals through that pathway, driving cancer risk and progression. Another clear picture emerged about inflammation, and so a number of adipose-derived and pro-inflammatory factors resulting from the increased adipose and dysregulated adipose tissue, driving inflammation, pathways such as NF-kappa B, COX-2 implicated in this process, and so clear evidence there, and also vascular effects, particularly through VEGF and driving increased angiogenesis and the like. So those were the established based on the weight of the evidence. There are also a number, well, I should include estradiol for the hormone-dependent cancers as well, increased aromatization, a clear picture there. There were some emerging mechanisms as well, epigenetic reprogramming, including DNA methylation, histone modifications, microRNAs, altered by obesity, and certainly emerging as a picture here that we're getting epigenetic regulation in response to the obesity of some of these cancer-related signals. Immunosuppression also emerging. Maybe today, if we looked a fresh look, we may put this into the established category. Clearly, obesity's driving an immunosuppressed state, increased T-cell exhaustion, exclusion of T cells, myeloid-derived suppressor cell story emerging. And so that seems to be an important part of the picture as well. There's also a story that emerged about metabolic tone, an interface between the systemic changes associated with obesity and intrinsic cell metabolism that we link with cancer. So the Warburg type of metabolism and driving down a glycolytic pathway, clearly affected by some of these systemic factors that are obesity related. So that's an emerging one. But our topic today, and also in the emerging category, is the microbiome. So I put a question mark there, and I think we'll get some answers, maybe moving closer to a clear picture on this. There are some key questions I think we'll be addressing today that our distinguished panel will give us some clear views on. One is obesity associated with changes in microbial composition? And the answer, I think, is yes for gut. It was even when we looked in 2018 or so. It's less clear for other microbiomes, and that's where some of the leading edge work is happening in breast, endometrial, prostate, for example. But yes, clearly obesity is changing the relative abundance of a number of bacterial genera, and that's a relatively clear picture now. Have obesity-related changes in gut microbiome been associated with changes in cancer-related pathways? Well, that's a yes. Multiple cancer hallmarks have been implicated. We see a number of pathways: inflammation, angiogenesis, clear tissue invasion, and metastatic processes impacted by, or certainly associated with, changes in the microbiome. There's deregulated metabolism. And the ability to evade the immune system appears to be also influenced by the microbiome. A third question: have microbial-derived factors been associated with pro-cancer effects of obesity? Well, yes, we're going to hear a lot about this today. This includes short-chain fatty acids, secondary bile acids, LPS from gram-negative bacteria, particularly polyamines, all involved, in some of these cancer-related pathways. Particularly inflammation has emerged clearly. So we've circled the wagons. We've got a lot of evidence, and we'll hear more today. Here's where I think the issues lie in terms of are we quite there yet? We've got more work to do for sure, and that relates to the causal links between obesity, the microbiome, and cancer. Have these been clearly established? And this is where a critical gap is I think still needing to be addressed, and we'll hear more about approaches to this. And then a fifth question: have causal links so if we're working through Koch's postulates, if you will, the other side of this is, well, what if we reversed the pro-cancer effects of obesity with various weight loss interventions, for example? Does that alter the microbiome? Can we get evidence there? And has this causal connection been established? And again, this is unclear. My group has been focusing a bit on this last question. As I said, the clearest pictures from bariatric surgery in terms of the human data as a way to reverse the obesity effects and prevent subsequent cancer risk down the road. All cancers, about a third reduction. When you look at the obesity-associated cancers, you see about a 40 to 50% reduction in risk following bariatric surgery. These are in morbidly obese patients. And a number of meta-analyses and systematic reviews on this clearly showing this is one way to do it. Now we're not going to operate ourselves out of this obesity cancer problem. So what is underlying this effect is a key question, and can we mimic it with other dietary pharmacologic approaches and get there as well? So we've taken a look at this in a number of model systems now. This is from one of our triple-negative breast cancer models, where we compared continuous control, continuous obese, obese for 17 weeks and then switched to a low-fat diet, obese for 17 weeks, then switched to a low-fat calorie-restricted diet, or a high-fat calorie-restricted diet, or an intermittent calorie restriction diet. And the intermittent calorie restriction approach we took was the 5-2 type of intermittent diet, where 5 days of a healthy kind of Mediterranean style diet, 2 days of a very low calorie, low carbohydrate diet. And we compared that to bariatric surgery, in our case, a vertical sleeve gastrectomy. So about a 70% reduction in the stomach size, turning the stomach from a pouch to a sleeve, hence the name. And that was, was effective. It reversed the pro-cancer effects of obesity here that we see, about a doubling in the obese compared to the control. The VSG brought it back to the control level. Interestingly, the diets, the restricted diets, were even more effective in terms of anti-cancer effect. And it didn't seem to matter whether it was a high fat or a low fat, a little more effective with the low fat calorie restriction regimen. And that intermittent, which people can do, it's not as onerous as a constant calorie restriction, was even more effective than that high fat calorie restricted diet. So some promising effects. One thing we have looked at a number of omic approaches with this, and from the secretome, what emerged is that leptin adiponectin ratio, really strong predictor of the anti-cancer effects. Now it's becoming clear that both leptin and adiponectin can have some regulation from the microbiota and some of the products of microbiome. So that's one interesting piece. We also did look at the microbiome using 16S RNA sequencing and some downstream approaches. And what's clear when we look at the top 10 of the bariatric surgery shown here versus the control, what were the top 10 differentially abundant bacteria? We see Akkermansia particularly going up with the surgery. This has been shown in human data relative to the control. The Hungatella as well, and Lactococcus also changing dramatically in comparison to the control. What was interesting is even though the restricted diets were fairly comparable in their, even a little better in their anti-cancer effects, we see a very different profile. They have their own signatures really of bacteria. So a lot more to learn here, but it is clear that these anti-cancer interventions are moving the microbiota. We're in the process of doing some sequel microbiota transplants to try to get to that causal question, but a lot more to learn. And I'm so looking forward to the panel today. So I will stop here and turn it over to our first speaker, and actually back to Phil to introduce our first speaker. So, and really looking forward to the session. Thank you.
Thank you so much, Steve, for that really outstanding overview and just covering some of the emerging areas where obesity and microbiome are overlapping. So there's this reprogramming of metabolic, epigenetic, immune, and then I would also add microbiome
Yes.
into that our obesity caused.
Yep.
I want to pause here just a second because I'm not sure if our participants were able to hear much of the introduction here, but we have set up the webinar today to field questions after each of our speakers talks very briefly, and then a larger panel discussion at the end. So I encourage all the participants, if you have questions, to use the Q&A box that has been set up. And that will be moderated by Dr. Sharon Ross at NCI, the question and answer. Okay. Just wanted to make sure everybody understood the webinar's format. Okay. So our next speaker is Dr. Christian Jobin, a professor of medicine at the University of Florida in Gainesville. Christian's also the co-director of the Cancer Therapeutics Host Response Program at the Florida Health Cancer Center. Dr. Jobin's research focuses on establishing a functional impact of bacteria in inflammation and colon carcinogenesis and deciphering their mechanisms of action. And his talk today is entitled Obesity-Associated Bacteriome and Development of Colorectal Cancer. How does it work? Christian?
Thank you, Phil, and welcome everyone. I'm extremely thrilled to share this symposium with you, and I'd like to thank Dr. Hursting for this outstanding introduction that really covered the material that we're going to go into more detail today. So, okay, we want to move forward. Am I on my slide? I'm not on my slide. Okay. This is the title. We're good. All right. So This is a tree that shows intestinal microbiota composition between case and control is different for different pathology, including obesity. And an interesting fact is obesity-associated pathology are also showing different microbiome pathology like metabolic syndrome, diabetes, hypertension, systemic inflammation. So it seems to be some sort of a pattern of microbial changes when you have obesity and obesity-associated pathology. Now, the next slide is showing you, you can't read that. This is a list of studies, 28 studies, so table-wise, that shows that they've been studying microbiome composition in case and control obesity or normal. So you have rich data set out there, and you could poke this and ask different questions. One question would be, is the fecal microbiome predictive obesity? If you start analyzing the microbiome, will you say this is coming from an obese patient? And one way to answer that question is to do the simple, relatively simple machine learning, some sort of artificial intelligence ways to look at a complex data set. And what you could see here is that the predictive value of microbiome is very poor for obesity. You could see that the purple or kind of magenta color here, compared to cirrhosis, for example, very high predictivity, colorectal cancer, or IBD. So not really predictive of the disease. Does it mean that it's the end of the game? Absolutely not. Traditionally, the microbiome of obese people have been characterized by a high ratio of Firmicutes, Bacteroides, with specific taxa, discrete set of taxa that associate with case, including Escherichia coli. I will come to that later. Now, it's important to mention also that the microbiome is present in tissue impacted by obesity, such as omental, mesenteric, or subcutaneous adipose tissue. For example, obese patients that end up developing type 2 diabetes show a decreased diversity of microbiome with an increased prevalence of enterobacteriaceae, and if you look at the genus level, Escherichia and Shigella. So there's different compartment, not only the gut, but also tissue-associated microbiome that has a difference of composition, and that may be associated with disease phenotype. Now, this is kind of a cartoon that incorporates important fact about the microbiome. If you think the microbiome as an organ, it could be injured. It could be injured through, I just mentioned that, inflammation. And Dr. Hursting mentioned the component of inflammation, obesity, and potential cancer. Obviously, diet, high-fat diet and with high sugar, low fibers, combined with a limited offset of let's say lifestyle, exercise, and so on, put you at high risk developing colorectal cancer and also obesity. Speaking of, if you look at data set, this is a study published by the group of Steve O'Keefe in Pittsburgh. You look at the risk ratio to develop colorectal cancer in a population from Alaska native versus rural African. The risk is almost 20 times higher. And these folks have a very different dietary intake, high fat intake, very low fibers, and also a rich or predominant protein from animal. And this dietary intake has a profound impact on their microbiome. If you look from the African, rural African versus Alaska native, you see a different microbiome. And when you look at the signal driving these differences, you will notice some taxa here listed, including the Escherichia Shigella signal that I've been mentioning in other form or group. So you could put this together. We talked about obesity, interaction with microbiota. How will this push forward your risk of developing colorectal cancer? One integrating factor between obesity and microbiota is likely the inflammation that could set the system in motion. It's a very important, inflammation is a very important component of microbial injury. and also as a factor of or a physiological response to obesity, this inflammatory component. So I put together this cartoon. It's relatively simple, but it shows that the host, if you think about, let's say, the intestine, it will be immune cells, mesenchymal cells, epithelial cells, and they interact with your microbes from, decades of life, from birth to the end of life. And this interaction is considered healthy up until you have exposure to modifiers. And these modifier, I mentioned to you, inflammation is a very important modifier. Diet, medication, and some component of genes may be impacting that. And as a result of this injury, this microbiome doesn't have the same configuration, the planktonic microbiome, but also the microbial organization on your tissue are likely to change. And we are on the opinion that these changes could lead to the formation of a carcinogenic network driven by microbes that promote neoplastic changes. And this network could be parsed in different elements. Here I list a couple of genotoxin and toxin coming from microbes that are important. I will highlight this one, colibactin, that comes from some species of Escherichia coli that is regularly found in obese data set. So if inflammation is indeed a very important component driving this change of microbiome, one question will be, is it possible plastically, in a plastic fashion, targeting the inflammation and then modifying this microbiome so you prevent this carcinogenic network from being active or being formed and active and promoting cancer. So we went into this to try to solve this relationship of inflammation, cancer. And we use an AP-Semin model. This is a model of small bowel polyps. But if you triggered epithelial injury with a chemical named DSS, then the phenotype will be seen in the colon. So you have colon cancer. And we use an E. coli that has this genotoxin I was mentioning, and treat this cohort or not with the anti-inflammatory molecule. In this case here is an anti-TNF, very important inflammatory molecule. And if you do that, you look at the cohort, you could see a decreased colorectal formation when the mouse has received anti-TNF. And you could see the polyps here been decreased in treated mouse. And if you look at the microbiome of these mice now, so the relationship between inflammation and the change of microbiome, you could see that the red and the blue here indicate microbiome of the individual mouse. And without treatment, there's no differences because they are the same genetic background. As soon as you start the treatment, day 14 is 3 injection of TNF. You could see a separation between red and blue. 35 is 6 injection of the anti-inflammatory molecule, nice separation. And on the tissue, once you harvest the mouse, you look at the microbiome in the tissue, there's also a differences. So very impactful differences in these mice in terms of microbiome. Now, an important component of microbiome is the transfer between mice. You could do an experiment to see the functionality of these changes. So if you co-house mice, they are coprophagic, they will transfer passively their microbiome. So now the TNF treated and the non-treated mouse will be sharing the same cage and exchange biome. If you do that experiment, it's the same treatment again. If you do this experiment, the anti-TNF treatment lost its effect. So the only thing that changed in this experiment is the access of microbiome from a non-treated mouse, which suggests the microbiome has a very important effect on driving the anti-inflammation and anti-cancer effect of an inflammatory molecule. And look at the red and blue microbiome. Now, before I show you the separation, there's no separation anymore if the mice are co-housed. So the microbiome passively transfer and prevent the anti-TNF effect. So the microbiome is very important. Functional impact of this. So this experiment was to address what does it mean if you change the microbiome? Is that preventive of cancer. And a way to do that is to harvest biome from an untreated mouse or the mouse that was treated with the anti-inflammatory molecule and put them in a new cohort that never saw treatment. So there's no treatment in that cohort. Just the microbes were coming from a treated mouse, and do the same model. And what you could see here, if you do this experiment, is that the microbiome lost its carcinogenic effect. So this is the biome of a treated mouse put in a fresh cohort, and you have the mouse has less propensity to develop colorectal cancer than a mouse a microbiome that was coming from an untreated mouse. So there is an impact of the treatment on the biome, and this is illustrated here. You could see that inflammation will change the biome. If you intervene on the inflammation with a molecule such as anti-TNF, for example, you collapse these changes, and then you reverse or you prevent neoplastic changes. So the microbiome is plastic. There's a plasticity and will respond to treatment and help prevent colorectal cancer. So how do we put this in the obesity context? What do we need? What will be the next step? And Steve mentioned that we need clarity on the characteristic of the obese-associated microbiome. What is the functional impact of obese-associated microbiota? And how plastic is the obese microbiota? The data I've shown you is by interacting on the inflammation. What is the obese microbes capable of? One way to do that will be to have access to feces from obese patients compared to a lean subject and isolate them through different ways. People use culturomic. This is a machine called Prospector that could be high throughput identification of isolate. You have a single isolate from a complex community that you could identify by mass spec or sequencing. So you create or reverse engineer an obese microbiome. And you could characterize this biome using mini bioreactor, where you could culture in continuous flow these complex microbiome and feed them with nutrients that you think may be deleterious to their activity or beneficial to reverse the deleterious effect of microbes. And you could measure these effects, by metabolite or different ways from the supernatant of this continuous culture. So this is more microbiology. The functional impact could be tested with access to lean feces or tissue from the gut, the same with obese, extract the microbes from them and input them into your favorite model of colorectal cancer and genomically characterize them to get clarity about who is driving the disease coming from a human perspective. And finally, how plastic is the obese microbiome? Steve mentioned that at the beginning. Is that fixed in time, or could you intervene through either diet or different means? And this is a study enrollment here, the BGON at MD Anderson, where participant, the idea is to target the gut microbiome of high-risk patient to mitigate the metabolic and inflammatory effect of adiposity that influence colorectal cancer. So you will try with, these folks are trying dry beans, try to modulate microbiome composition and prevent risk factor for colorectal cancer. And the completion of the study will be in June 2025. It will be interesting to see how they will perform on that task. But one thing that is interesting to mention is that sometimes change in microbiome may not be happening, but yet you have beneficial impact. For example, the microbiota of severe obese patients subjected to Roux-en-X gastric bypass don't gain diversity. It still remain at low diversity, but yet the bioclinical improvement, metabolic function improve with the surgery. So it didn't parallel the microbiome changes. So there's a little bit of complexity, change by composition versus change by function. So there's a lot to learn still at this point, but you could see that from the webinar we're going to have today, there is a big gain of knowledge that is coming in this field of research. I'll stop here and just have this slide to illustrate or acknowledge the people who participate in this study. And I'll end it here. Thank you.
Thank you, Dr. Jobin. Very nice presentation and getting us up to speed a little bit on the plasticity of the microbiome and obesity. I'm curious about when you talk about the obese microbiome, you're mostly, my understanding is you're talking about the fecal microbiome. And I'm wondering how much the adipose tissue
Yes.
microbiome
Absolutely. Right.
contribute to that. And I know you study that. Could you comment a little?
Right. I mean, so the obesity, the tissue we had, the deposited tissue was coming from colitis, Crohn's disease patient, right? Not obese patient, but their study have been done with patients that have type 2 diabetes, hypertension, and that are obese, and they found in these adipose tissue presence of microbiome. We're not quite sure what is the function of them in terms of disease outcome. I mean, do they participate in the systemic inflammation? Do they, promote the obesity secondary disorder? So not 100% sure what's happening. For colorectal cancer, at least for IBD, these microbes seems to have, if you resect the tissue, the colon, but don't take care of the mesenteric fat, you relapse into inflammation within 5 years. So they hide there. They are hiding in plain sight. And even if you remove the inflamed tissue, these bugs are there and come back and promote inflammation. So it's kind of you could almost view that as a reservoir somehow. And I'm not sure if it will be the same for, obesity or type 2 diabetes and so on. There's no surgery per se on type 2 diabetes, but you will think that these microbes could participate in the systemic or pathology associated with obesity.
So a lot more to understand. One more question before we go to the next speaker and Phil's introduction. Someone asked, would you please recommend anti-inflammatory foods and supplements for assisting with gut health and anti-inflammation? Maybe we can also cover this a little bit.
Yeah, that is
during the panel from everyone.
This is a discussion. I think we could put a mark on that question and come back to it. But I think it's a very important question. So let's earmark it, if you don't mind. We'll come back.
Sounds great. We'll come back to that, and I'll hand it over to Phil for now. Thank you very much for a nice discussion.
Absolutely.
Okay, thank you, Sharon, and thank you, Christian, for that very interesting talk. And again, I want to remind everyone, if we didn't get to your question that you posted in the Q&A box right now, we only have time for one or two after each of the talks. We will come back to it during the panel discussion at the end of the webinar. So hold tight for those answers. Okay. I'd like to introduce our next speaker, Dr. Liza Makowski. Dr. Makowski is professor of medicine at the University of Tennessee Health Science Center in Memphis. Her lab focuses on immunotherapy and immunometabolism and on reprogramming of the immunosuppressive tumor microenvironment, microbially derived metabolites, and the efficacy of immune checkpoint blockade therapies in obesity-associated, obesity-induced breast cancer. The title of her talk today is Obesity, Weight Loss, and Immunotherapy in Triple-Negative Breast Cancer. Dr. Makowski. Liza, I'm not sure you're unmuted.
Now, can you hear me now?
Yes.
Okay. I don't see where I have control anymore to turn on my screen now that I have control. Can you guys do
Yeah, there seems to be a bit of a lag, but I think if you keep trying to advance, I think eventually it will.
Okay, can you hear me now?
Yes.
Okay.
Liza, we will just take momentarily slide control back so you can turn on your video and pass it right back to you.
Perfect. Thank you. Yes. Sorry. Tell me when. Yes, here we go. My video is on. Now I will take control. Got it? Great. So now I think I have control. Perfect. Okay. Thank you, Dr. Dashner and Dr. Ross and everyone for inviting me to this webinar today. It's really exciting. I think we're all going to learn a lot from each other and during our discussion. So today I'm going to talk about obesity and immunotherapy in breast cancer. And I am, one second. As Steve Hursting alluded to in the beginning, we're really interested in obesity and cancer. And not quite a decade ago, ASCO, the American Society of Clinical Oncologists, put out a white paper saying that we need to start paying attention more to obesity and cancer, not just for prevention and risk, but also because it impacts care and outcomes. So this has become a real priority in the cancer world and kind of integrating more with obesity researchers. And so how does obesity impact breast cancer? So it negatively impacts many breast cancer outcomes, from local invasion to distant metastases to recurrence following treatment. And obesity leads to all these things together culminate in increased mortality, both in postmenopausal women and especially in premenopausal women. And so when we think about obesity in the United States, and we go to these awesome slides provided by the CDC, where they look at statistics for obesity as looking at prevalence of BMI. So body mass index over 30 is what we indicate for obesity. And the darker the color from green, yellow to maroon, we see increasing prevalence of obesity. But if you, instead of just looking at the United States as a whole, we break it out by racial and ethnic composition, we see a smattering of states with white non-Hispanics where there's high prevalence in maroon of obesity, much more with Hispanic, especially in the center of the country. But what is striking is the number of states with very high prevalence in Black non-Hispanics. And we'll get to why that's really important in a second when we talk about not just breast cancer, but breast cancer subtypes. So breast cancer is, like many other cancers, defined by different subtypes, which have different clinical risk factors and outcomes. And if we look at some of the intrinsic subtypes, and in a little cartoon borrowed from Melissa Troster, we could look at the size of the circle representing about prevalence. So the bigger the circle, the more prevalent. So we know that the ER positive type cancers, like the luminal cancers, are quite prevalent in populations, whereas triple negative breast cancers like Claudin-Low and basal-like are have much lower prevalence. And so why is this important? Well, because despite being lower prevalence, they actually have very poor prognoses and very few treatment options because they are negative for receptors that are often targeted by therapeutics like ER and PR and HER2. And so if you look at, like, for luminal A, the dark blue line, this data is a little bit old, but we could get the point that often these patients have much greater survival compared to patients with triple negative breast cancer who have poor outcomes. And so how does this link back to obesity? Well, triple negative breast cancer, as I mentioned, has very poor prognoses because of these fewer treatment options, but it also can disproportionately affect young and minority women, both African American and Hispanic. So this really can drive home the need to increase potential therapeutics, as well as to help impact health disparities. Because if we can be reminded, non-Hispanic, Black women especially, are very impacted by obesity. And so what I'm really interested in is the role of the microenvironment and how cells talk to other cells. And there's a really great person who's really a pioneer in the field and thinking and talking about how the soil can impact the seed. So Mina Bissell was a real pioneer in looking at how the microenvironment or the soil around the cancer cell or the seed can impact that cancer growth. And this is a cartoon from one of their reviews, where if we look at the role of the microenvironment in tumorigenesis over time, we see a real dichotomy or dual role of the microenvironment. So early on in the breast or the mammary gland, we have a bunch of these cells all contributing, as well as extracellular matrix, etc. And that's actually suppressive to tumor formation. So there's this barrier that kind of does not promote progression. But you cross this threshold, and soon the microenvironment changes to where it actually becomes promoting and pushes that microenvironment towards tumorigenesis to increase and promote tumor formation. And so, as Dr. Hursting alluded to, how does obesity play a role in this? Well, we know that it can strongly promote changes in the microenvironment and tumor progression. And so today I'm going to talk about the role of immunosuppressive ligands and immune cells in obesity, and how we can see, as Dr. Hursting mentioned, T cell exhaustion and some changes in immunosuppressive cells. So I'll briefly introduce what are immune checkpoint blockades. So these are the ways cells talk to each other. So when we have an antigen presenting cell like a dendritic cell, it's going to communicate to a T cell to activate that T cell to induce a response. But we don't want a hyperreactive response. We need to tone down a response after it happens. So there's signals to deactivate that T cell, such as you've probably heard of PD-L1 or PD-1, and these all act to then tune down that inflammation. And what happens in cancer is that cancer cells, the tumor cells, hijack this, and they express ligands that crosstalk to T cells to tune down that T cell and basically make it immunosuppressed. And in this way, your cancer cell can hide and evade the immune system. And so, of course, in the past decade, there's been real revolution in ways to modulate the immune system in cancer, and ways to block these immune checkpoint proteins with antibodies. So for example, we could break this crosstalk between that cancer cell and the T cell by using antibodies that block the two cells communicating. And that then reinvigorates your T cell, gets it ready to then attack the tumor cell and kill it. There's lots of different antibodies out there and other ways to reinvigorate your immune system, but today I'm going to talk about anti-PD-1. And so what's also really interesting to us in the obesity field is that it's not just a tumor cell communicating with a T cell through these ligands and receptors, but other cells in the microenvironment, which we know are changed or modulated by obesity or metabolism, such as dendritic cells, macrophages, myeloid-derived suppressor cells, or MDSCs, which I'll talk about in a minute, or even adipocytes, can express these ligands and be regulated by obesity. And so there's real interesting work coming out on the role of how these cells can either interfere or interact or improve immune checkpoint blockade. And so, as I mentioned, immune checkpoint blockade and immunotherapy has really revolutionized cancer treatment and has been highly effective and exciting in diseases like melanoma, lung, etc. It's been somewhat effective, but less effective in populations of breast cancer or triple negative breast cancer. And so let's think about what's happening with obesity and immune checkpoint blockade, and how might we improve efficacy in certain populations that especially need it. So again, we've got our antigen presenting cell like a dendritic cell communicating to a T cell, but this is a T cell in the lean state. This is an anti-tumor CD8 positive T cell. These are effector cells that are going to kill the tumor cell. And when they're lean, they might express some of this PD-1. But what happens in obesity, Wang et al. Have shown that when you're obese, and increased adiposity increases leptin release and signaling through transcription factors like STAT3 to increase PD-1 on that T cell, so that your T cell in the obese state is then prone to be exhausted. And other groups have also shown that cytokines, like we just heard about from Dr. Jobin, TNF-alpha, which we know is increased in obesity, can also regulate ligands even through protein stability. So we're learning many different ways that obesity can regulate immunosuppression. But is that good or bad news? Because while obesity can regulate immunosuppression and cause exhaustion, there's recent data showing in patients that obesity can improve immunotherapy in melanoma and other cancers. So this great review, oh, sorry, let me introduce an example of that first. Here's an example of metastatic melanoma treated with different immunotherapies, and they pooled the cohorts by body mass index. So normal weight patients in that are lean in red, they have poor survival over time. And when you look at obese or even overweight patients, they have improved survival with immunotherapy. So this is really exciting. And this is a review that came out in 2020 looking at improved survival, overall survival, and progression-free survival in a bunch of different cancers, as I mentioned, melanoma, renal, non-small cell lung cancer. But what about breast cancer? That data is not quite clear yet. And so here is where we were able to take advantage of our preclinical models and ask these questions that we can manipulate a little bit better than a patient population, and look at the role of immune checkpoint blockade, how we can reprogram local and systemic immunity. And this is led by Dr. Pingley and my current grad student, Mehdi Shaib. So what I'll tell you today, we looked at lean and obese models, and we showed that in cases where obesity has exacerbated tumor burden, we can reverse that with immunotherapy. So we use C57 Black 6 model, which is a highly obesogenic model, and feed them these diets which are matched and defined from research diets. And despite being female mice, we did not do any ovarectomy. They got quite obese, as I'll show you. And what's really important is that these mice are immune competent. So we're importantly looking at immune interactions in this model. And we inject a C57-derived syngeneic cell line, the EO771s, which are orthotopically injected into the mammary gland so that we could ask the questions, what's happening with obesity locally and systemically? And so here's our little cartoon of our mouse model, and we inject the cancer cells into mice that are already lean or obese. So these mice are fed the diets post-pubertal at 8 weeks until basically middle age. And you could see from lean to obese mice by body weight, or lean to obese when we use MRI to quantify adiposity. These mice are quite obese. And then we inject the cancer cells to model progression and the impact of immunotherapy on our varied outcomes. So what happens? So as Dr. Hursting alluded to, and our group has shown, other groups have shown that obesity can drive tumor progression in orthotopic or transgenic models of cancer. And same as what we see here, these are the cells in obese that are quite rapidly progressing. And interestingly, we didn't see a great progression in our lean models. And if we break it out, and if we look at the response to immunotherapy in red, we see that this obesity-driven tumor progression can be reversed by anti-PD-1. And so we see this reduction basically down to lean levels. And if we break it out by individual mice, and this is looking at progression over time in individual mice, we see a real reduction of progression. And even if we look at regression, so palpating at endpoint, we see that 4 out of 12 regressed with immunotherapy, but no regressed in our obese mouse. Likewise, in the lean, we didn't see a lot of tumor progression in our hands, but 8 of the 9 tumors regressed with immunotherapy, even in the lean state. And so just to summarize this, we saw that this obesity-induced progression in the black spots can be dramatically and significantly reversed by anti-PD-1 immunotherapy. And so what happens, why is this happening? And we looked at first obvious things like immune cells in the tumor microenvironment, but also systemically to see how obesity impacted this. And I'll tell you about some changes obesity-induced induction of immunosuppressive cells, and then how we reverse those with anti-PD-1 immunotherapy, and also drove the increase of anti-tumor cells. So here's just an example where if we look at our obese tumors, we did both flow and immunofluorescence for T cells. These are CD3 positive cells. they're increased with immunotherapy as expected. And these are primarily CD8 positive driven changes in our immune tumor microenvironment. And likewise, when we did RNA-seq analysis, we could look at other cells by informatics using timer point 2.0. And so cells we didn't do flow for, but for example, CD4 positive Th1 cells have increased from blue to yellow, as well as the overall immune score. So we've reinvigorated immunity by decreasing these immunosuppressive cells and increasing these anti-tumor cells. And so what's really interesting is that in the paper, I'm not showing everything, but we see that we could reprogram M2 macrophages, myeloid-derived suppressor cells in lean and obese tumor-bearing mice with immunotherapy. But when we compare that to mice that are tumor-free, we were able to look at lean and obese mice without the tumor, and we saw changes that were present only when tumor was on board, not in the absence of tumor. So for example, myeloid-derived suppressor cells, both the PMN and monocytic myeloid-derived suppressor cells, were not really changed with obesity, but were dramatically changed in the presence of tumor. So the take-home point is that this obesity-driven immunosuppressive priming is really dependent on the presence of the tumor and crosstalk. And so I've mentioned clinical studies and a little bit of our data showing that there's this paradoxical yet serendipitous event where we see increase in immunosuppressive cells and immune checkpoint proteins that lead to better immune checkpoint blockade outcomes. But what might be mediating this? And this is where I'm here to show some data and learn today about the gut microbiome and how that can affect immune cell function. So the gut microbiome we know can change in terms of overall content, composition, and the secretion of antimicrobial peptides or metabolites, etc. And these can act locally as well as in distant sites to impact immune cell biology. And so we took our models, we took advantage of the models that were treated with control or immunotherapy, as well as tumor-free models, and looked at the gut components in two different regions, the cecum and the jejunum, and looked at how immunotherapy impacted that. Now in the paper, we've included a lot of low-fat and high-fat changes, which were as expected. But here I'm just going to talk about our obese focused group, where we have non-tumor or NT, the IgG or the anti-PD-1 immunotherapy treated. And you could look at jejunum or cecum and see that obviously these are two different depots in the gut, and they have very different profiles. But we could also then look, how are these changed differently by lean or obese or immunotherapy? And so I pulled out some ones that were really interesting to me. These are cecal microbes that are either in purple, non-tumor, and here they're more high, and here there's no expressed. But when we compare that to when tumor is on board, in tumor-bearing mice in orange, compared to the immunotherapy-treated mice with tumor, we see this reversal of this microbe ruminococcus. But then here, there's almost no Bacteroidealis present, but when tumor's on board, we see this increase, which is reversed with PD-1. Likewise, we see some where there's no changes when tumor's on board, but a unique increase with anti-PD-1 immunotherapy. And so these are things that we're further investigating. And we then took a step back and say, which one of these microbes can correlate with our tumors? And we did lots of correlation and regression studies and found one that was really interesting, that with increasing content of, this is again, cecal gut Enterobacteriaceae, we see increasing content relating with tumor size. So the larger content of Enterobacteriaceae, the larger the tumor. And interestingly, this is downregulated with immunotherapy. So we were excited about that, and we have created what we call an immune checkpoint blockade associated signature. Of course, we don't know if this is causal or maybe a putative biomarker, but at least in this model, we see in obesity-associated breast cancer, in the presence of a tumor, we see some downregulation and upregulation of certain microbes. And with anti-PD-1 immunotherapy, we see these changes in the gut with an increase in these microbes and a decrease in these microbes. So I just mentioned Enterobacteriaceae. And so when I went back and I looked at one of our collaborators, this Catherine Cook is at Wake Forest. She's a co-investigator on our R01. She published in 2019 something that really struck our interest. So she has patient samples that are lean or obese and looked at tumor, breast tumor microbial content. So I really recommend you to look at this paper. And in these tumors, she sees a decrease in Ruminococcaceae and an increase in Enterobacteriaceae. And if you remember, we saw the opposite with immunotherapy. So where this is low in obese tumors in patients compared to non-obese patients, this was higher in our gut. And the reverse is true for Enterobacteriaceae. So this is really exciting. And of course, as Dr. Hursting alluded to, we see these changes, but what can we do? How can we reduce? And as he mentioned, bariatric surgery is associated with reduced incidence. But it's really not clear how we're getting this protection and survival in patients who've had bariatric surgery compared to controls. And so we've also been interested in looking at surgery and diet intervention. And we have a paper in submission led by Dr. Laura Sipe, a fellow in my lab, and contributed to by my grad student, where we look at surgically induced weight loss and compare that to different diet interventions and the role of immunotherapy. And I'm excited to say that Laura just received an F32 for this project. And so I'll just end by saying that as part of our R01, we're also interested, of course, in looking at how weight loss, such as bariatric surgery or medically induced weight loss, can and changes in the microbiome can impact cancer. So through myself and especially Joe Pierre, our MPI, who led the work getting an IRB approved during COVID times, with our surgeon, Matthew Davis at the Weight Management Wellness Clinic, we have been able to start collecting patient samples, fecal samples for future study. So I just want to end by saying I acknowledge my collaborators throughout and my lab, students, and our funding. So thank you very much.
Thank you, Dr. Makowski. Very interesting. Thanks for all your telling us about all your work on obesity's relationship with immunosuppression and immunotherapy and the microbial signature that you found in the animal studies as well. We have a question going back to some of the human studies. For the studies showing that higher obesity is associated with better survival, what is the comparison group? And if compared to lean patients who may be lean because they have more aggressive disease and losing weight. Sure, you can go ahead. I think we got I had some incoming email that was blocking the questions, but I think you got the gist of it. Thanks.
Yeah, I heard you. I mean, I think also this is something we might be able to talk about during the panel, because of course these weren't my studies, but we would have comparison groups that would be healthy, lean controls. And I think we should really talk about this during the panel, because when you think about obesity and weight and patients presenting with cancer, we have to think about, have they lost weight? Just this, exactly what that question's asking. So can we table that to the panel?
Yes, yeah, let's do that. No problem. Probably, I have another one probably that may or may not be tabled. I was interested in, you know, in some of these studies, the human studies that you were reviewing, whether or not, in addition to BMI, did anyone look at visceral or subcutaneous adiposity, or, you know, getting at where the adiposity might be coming from, and how that may be influencing immune function.
Right. I think also that's a huge panel discussion question, because of course, when you're doing large scope or any sort of high throughput analysis or patient analysis, it's easy to do BMI because you could pull that right from the chart. It's a lot harder. And and my training has always been in nutrition and public health. And then the first 8 years of my career was in a nutrition public health department. So the question is getting to patients, waist hip ratio or other ways, DEXA, MRI, the bod pod. How are we going to do this and answer some of these questions? I'd love to talk about on the panel.
Great. I think perhaps we have to move on. And we have a couple other questions, but we can get to that at the panel as well for you.
Okay. Thank you, Sharon. And thank you very much, Liza. Very interesting talk. And yeah, I have a couple of questions too about the intratumoral microbiome and how that's affecting therapy. But Sharon's right. We'll get to that at the panel discussion and hear from everyone. But now I'd like to introduce our final speaker this afternoon, Dr. Matam Vijay-Kumar. Dr. Kumar is professor of physiology and pharmacology at the University of Toledo College of Medicine in Toledo, Ohio. His research is focused on host metabolic adaptations to inflammation, and specifically how the adaptation of alternate metabolic pathways that protect against inflammation can culminate in metabolic disease and cancer. And the title of his presentation today is Gut Microbiota Dysbiosis, Dietary Fermentable Fibers in a Pickle, a Brew for Liver Cancer. Dr. Kumar.
Thank you very much. First of all, I thank Dr. Dashner and the organizing committee for the invitation. So today I'm going to tell you a story of a mutant mice which took us to the liver cancer. So how can I do this? Presentation mode?
I think there's a lag. But...
Oh. Okay, can you see the slides?
We can see, and maybe you have control now.
Yes, we can see your title slide.
Okay. Yeah. So already the previous speakers, Dr. Jobin and Makowski, excellently introduced the gut microbiota. So I will keep my introduction very short. So far the bacteria belonging to 52 phyla have been identified on the earth, but only 5 to 7 phyla can colonize our gut, where they can form stable community and also productive ecosystem, thereby both the host and bacteria get benefited. So interestingly, even the proteobacteria dominates on the earth, but it is a minor community in all the mammalian guts. So gut microbiota can be regarded as a neglected organ in organ. So as you know, the mammalian intestines, which are lined by a single layer of epithelia, is also having a largest surface area, almost like a tennis court, which is also house for 10 to the power 14 bacteria, or zillions of bacteria belonging to 500 to 1,000 distinct species. So the major phyla in our guts are Bacteroidetes and Firmicutes. In fact, we carry more bacterial genes than our own genes, and the functions of the gut microbiota is comparable to that of liver because they can influence the distant organs metabolism like bone mineralization and also lipid composition of our islands. So in other words, the host and microbiota, they hold each other throughout the life, which starts at the birth and ends with our body decomposition. So we can regard our gut bacteria or microbiota as a best frenemy forever. So because majority of times they are very helpful to the host, but at some times they can cause moderate to serious cases, disorders. So it is a Herculean task for the host to maintain such a huge number of bacteria in the distal colon. So the gut has developed a specialized immune system in which pattern recognition receptor is one of the components which falls under innate immunity. So broadly, they can be classified into 3 different forms, like secreted form or transmembrane form and cytosolic form. So I'm not going into the details of all these. We are interested in membrane-bound form of receptor, that is toll-like receptor 5, abbreviated as TLR5. So the toll-like receptors, they sense microbial ligands, and they are highly conserved in the evolution. They are type 1 transmembrane proteins. So they recognize specific microbial ligands, for example, TLR2 peptidoglycan, TLR4 LPS, whereas receptor of our interest, TLR5, which is predominantly expressed on epithelial cells, including intestinal epithelia, it is a receptor for flagellant. So once they bind to the respective ligands, which results in the activation of a transcription factor, NF-kappa B, which enters the nucleus and initiates the transcription of variety of pro-inflammatory genes. This pro-inflammatory gene expression is required by the host because that will help in the getting rid of any invading pathogen. At the same time, this pro-inflammatory response should happen in a controlled fashion, otherwise it can result in unnecessary inflammation to the host tissues. So when I started my career in the Emory University, my goal was to study the interaction between the toll-like receptor 5 and flagellant in inflammatory bowel diseases. So the best way to study their interaction is using TLR5 knockout mice. So we obtained a TLR5 knockout mice from Dr. Akira, who is a pioneer in toll-like receptor research. And first we verified the loss of function of TLR5 in these mice by intraperitoneal administration of flagellant. So as you can see, the TLR5 knockout mice failed to induce any pro-inflammatory cytokines like IL-6 or KC or GCSF. So lack of such a pro-inflammatory cytokine response to flagellant, we hypothesized that TLR5 knockout mice would resist inflammatory bowel disease, at least in the murine models of colitis. Interesting, in contrast to our hypothesis, we observed spontaneous colitis in the TLR5 knockout mice. So approximately 10% of the mice, they develop rectal prolapse, which is a severe and irreversible form of colitis. And 30% of the mice had mild to robust colitis as analyzed by colomegaly, shrunken cecum, and splenomegaly. And remaining 60% of the mice are indistinguishable from the wild-type mice without any signs of inflammation at the histological level. The TLR5 knockout mice develop spontaneous colitis because they are unable to control their gut microbiota. So they have moderately elevated fecal bacteria and substantially elevated colonic adherent bacteria, and also their sterile organs such as spleen and liver, they have clearly detectable bacterial burden. They found to be Staphylococcus saprophyticus and hominis. These are not pathogens. These are commensals in the laboratory animals and also pets. So the spontaneous colitis in TLR5 knockout can be prevented by broad spectrum antibiotics, suggesting that it is completely microbiota dependent. So next we asked whether these mice may be harboring a opportunistic pathogen that could be driving pathogen colitis. So therefore we re-derived TLR5 knockout mice using Jackson female mice. So in other words, we Jacksonized these Japan mice by putting them into Jackson microbiota. So the re-derived offspring acquires microbiota of birth mother, but not the genetic mother. By re-derivation, we completely prevented rectal prolapse in these mice, but still the re-derived TLR5 knockout mice has a colomegaly, splenomegaly, and as cecal myeloperoxidase, an indicator of neutrophil infiltration, and also elevated general inflammatory marker lipocalin-2. So in other words, the TLR5 knockout mice suffers from low-grade inflammation, which is a major player in all the new age disorders, including obesity and type 2 diabetes. So what we surprisingly found was the TLR5 knockout mice are obese. Of course, their obesity is not as severe as in leptin knockout mice, but one can easily find out by simply taking their body weights. This obesity is with the lab chow, not with high-fat diet. So we thoroughly characterize the obesity phenotype in the TLR5 knockout mice. As you can see, both males and females, they suffer from obesity. They are almost 20 to 25% more body weight than their wild-type litter mates. The TLR5 knockout mice also have had elevated serum triglycerides, serum cholesterol, and MRI scanning revealed increased fat distribution throughout the body. Their abdominal fat pads weigh twofold higher than the wild-type litter mates. When we measured their blood pressure via tail cuff method, they are clearly hypertensive. So collectively, all these results suggest that TLR5 knockout mice suffers from metabolic syndrome. Next, we focused on one of the major features of the metabolic syndrome, that is insulin resistance. So as expected, that obese TLR5 knockout mice are hyperglycemic, even though they are hyperinsulinemic. They also have reduced insulin sensitivity, so suggesting that they suffer from insulin resistance. So, which is a major feature of metabolic syndrome. When they challenged with the high-fat diet, it aggravated metabolic syndrome. So interestingly, what we found is the TLR5 knockout mice suffers from insulitis, which is characterized by new immune cell infiltration in the beta cells, and they also have severe hepatic steatosis, a major manifestation of metabolic syndrome. So obesity in the TLR5 knockout mice is because they are hyperphagic. The TLR5 knockout mice, they eat 10% more food per day, and accordingly, they have increased wet and dry stool output, suggesting that they are not having suffer from any diarrhea. Calorie restriction via pair feeding greatly improves metabolic syndrome, including hyperlipidemia in TLR5 knockout mice. So when we profiled their microbiota, what we found is that we found only the taxonomical alterations in the TLR5 knockout mice. They also have a reduced diversity, increased volatility, but no change in the major phyla as observed in leptin deficient mice. So finally, we rederived TLR5 knockout mice in the germ-free condition. So basically, the germ-free TLR5 knockout mice are completely free from colitis, low-grade inflammation, and also metabolic syndrome. So finally, we asked to what extent the TLR5 knockout microbiota is sufficient to transfer metabolic syndrome. So here we simply took the cecal contents from the wild type and TLR5 knockout mice and orally gavaged into wild type germ-free mice. As you can see, the germ-free wild type mice, which were given TLR5 knockout microbiota, they are hyperphagic, they gain more body weight, they're hyperglycemic, hyperinsulinemic, and also they have reduced insulin sensitivity. They also have increased abdominal adiposity with increased pro-inflammatory cytokine expression in their colons. So we also showed that TLR5 knockout mice with their increased bacterial burden, they generate more short-chain fatty acids, which and enter into the liver via enterohepatic circulation. They can participate in hepatic lipogenesis, and increasing the hepatic neutral lipids like triglycerides and cholesterol esters, which results in fatty liver that kicks in insulin resistance, and finally hallmarks of the metabolic syndrome. So when we deleted hepatic lipogenic enzyme, that is steroyl-CoA desaturase 1, we prevented majority of the indices of metabolic syndrome in the TLR5 deficient mice. So next we asked does dietary prebiotic fermentable fiber corrects or aggravates metabolic syndrome in the TLR5 knockout mice? So, so naturally occurring dietary fibers can be classified into either non-fermentable or fermentable fibers. So cellulose is the best example for non-fermentable fiber like cotton and paper, so which is a poly-D-glucose, whereas inulin is a polyfructose. Even though both appear like a white amorphous powders, their metabolism is chemically they are very distinct, and also their metabolism is different. Cellulose has little access to fermentation to our gut microbiota because our mammalian gut microbiota do not express cellulases except ruminants, whereas inulin can be readily fermentable by gut bacteria, resulting in millimolar levels of short-chain fatty acids like acetic acid, butyric acid, and propionic acid. So recently, FDA has approved to use inulin in the processed foods, including infant formula. So therefore, we thought of including inulin in the diets so that it can cure its dysbiosis and prevent metabolic syndrome. So we approached Research Diets vendor who has already an open source diet formula where they have 2.5% cellulose and 7.4% cellulose and 2.5% inulin. We simply ask them to reverse the fiber composition, keeping the total fiber composition to 10%. By feeding inulin containing diet, we prevented obesity in the TLR5 knockout mice. Almost 40% of the TLR5 knockout mice, they have reduced body weight and also abdominal adiposity. Their their blood glucose and insulin is comparable to that wild type littermates. And these mice which are protected from obesity, they have abnormally low levels of serum triglycerides and serum total cholesterol. So we were happy that by giving inulin diet, we prevented 40% of the obesity in 40% of the mice. But twist to the story happened when we were processing the serum samples. We observed yellow color fluorescence in the mice which are protected from the obesity. This happens within 2 weeks after inulin diet. And it could be because of the cholestasis. This is like a reduced blood bile flow or no bile flow at all. And this yellow color is due to increased bilirubin. So based on the bilirubin, we stratified TLR5 knockout mice into low bilirubin or high bilirubin. So we characterized the high bilirubin mice, like for alpha fetoprotein, a putative marker for liver dysfunction. So the TLR5 knockout mice with high bilirubin, they have elevated levels of alpha fetoprotein, and there is a nice correlation between the alpha fetoprotein and bilirubin. The high bilirubin mice also exhibited elevated levels of organ damage markers like serum alanine transaminase, aspartate transaminase, and alkaline phosphatase. So when we continued for 6 months on this inulin-containing diet, the mice with high bilirubin, they develop multinodular liver cancer. We extensively characterized these tumors histologically, and we found that they suffer from hepatocellular carcinoma, a form of deadly liver cancer. And they also have liver cancer specific markers like increased deposition of collagen, indicator of fibrosis, and reticulin staining, and elevated levels of proliferative cellular nuclear antigen cells. These are the black spots, which are highly proliferating hepatocytes. So this is the progression of liver cancer in the high bili mice. So at 1 month, you don't see any tumors. By 3 months, they suffer from steatohepatitis. By 6 months, the tumor starts appearing. By 9 months and 12 months, every lobe of the liver becomes tumorigenic, and finally the mice die because of hepatic encephalopathy. So next we asked, because inulin is fermentable by gut microbiota, so microbiota may be driving the liver cancer in these mice. So therefore we administered broad spectrum antibiotic in the drinking water that prevented liver cancer in the TLR5 knockout mice. And similarly, when the germ-free knockout mice were maintained on inulin-containing diet, they also failed to develop liver cancer. So suggesting that the liver cancer in the TLR5 knockout mice is microbiota dependent. And also we intervened with their fiber at the early time point. So when we got the high bilirubin mice, some of the mice were given switched to 100% cellulose fiber. By switching the fiber, we prevented the liver cancer in the TLR5 knockout mice, and also their bilirubin levels were reduced. So this is we thoroughly profiled their microbiota. So high bilirubin mice had 3-fold increase in their microbiota load, especially the mice which develop liver cancer. They are enriched with the Clostridia species. These are well-known fermenters and also producers of secondary bile acids. They also enriched with the proteobacteria, which are known to be opportunistic pathogens. So they also have increased fecal lipopolysaccharide, flagellan, and their serum is increased immunoreactivity to LPS and also flagellan. This is the Western blot showing increased immunoreactivity to E. coli proteins, suggesting that there is an increased dissemination of microbial products in the TLR5 knockout mice with liver cancer. So we also profiled the short-chain fatty acids in the cecum. So we didn't see much differences in the acetate and propionate, but whereas butyrate is substantially elevated in the TLR5 knockout mice. So as I mentioned, the bilirubin is one of the marker for cholestasis. Therefore, we also measured the serum bile acids, which are also marker for cholestasis. So as expected, the mice with high bilirubin, they have 20-fold increase in the bile acids. Those bile acids are modestly detectable in their feces. So suggesting that they are suffering from cholestatic liver cancer. As you know, the bile acids are very powerful detergents. They cause non-specific hepatocyte cell death. Accordingly, when we performed tunnel staining, you can see extensive hepatocyte death in the areas surrounding the portal triad. So, as I mentioned, the Clostridia species are very good for generators of secondary bile acids because they express a specific enzyme called bile salt hydrolase, which can deconjugate taurocolate into colate, which further acted upon by other bacteria, forming a potent hepatotoxic bile acid, that is deoxycholic acid. So accordingly, the mice with the high bilirubin, they have increased levels of bile salt hydrolase activity. So when we profiled their bile acids in the serum, every bile acids were elevated in the mice with liver cancer, specifically the deoxycholate also. And the same bile acids either not detectable or substantially reduced in the feces, again highlighting these mice suffer from severe cholestasis. So next we asked what kind of immune cells are driving the hepatic inflammation in the early stages of inulin diet feeding. So we extensively profiled immune cells in the liver. So what we found is by 4 weeks of inulin diet feeding, the livers exhibited elevated levels of neutrophils, and also they are positive for liver myeloperoxidase and elastase, which are exclusively expressed in the neutrophils. And neutrophils also very good sources for reactive oxygen species. They have elevated reactive oxygen species in the liver cancer mice, in the livers of cancer mice. And they also have increased levels of malondialdehyde, an end product of lipid peroxidation, which is a notorious aldehyde with 2 aldehyde groups, which can react to any amino groups either in the DNA or protein, forming a DNA adduct or skips base, which can cause DNA damage. So accordingly, what we found was elevated levels of 8-hydroxy-deoxyguanosine in the serum and urine of the mice which developed liver cancer. So what we think is, so far, the cholestatic liver cancer in the TLR5 knockout mice is fueled by deadly 3 Bs, that is the host and the bacteria-derived co-metabolites, that is bile acids, and the bacteria-derived butyrate and the host-derived bilirubin can drive the tumorigenesis in this TLR5 knockout mice. The reason is the bile acids-induced cell death and butyrate-induced cell proliferation is happening in the severe inflammatory conditions and also oxidative stress conditions that can drive DNA damage and tumorigenesis. So despite our conclusion is, despite the beneficial aspects of prebiotic fibers, the random enrichment in the processed foods should be approached with a great caution, as it may increase the risk of liver disease. So this I stop here. I thank my collaborators, Dr. Andrew Gevers, Andrew Patterson, Patrice Cunny, Jose Garcia, and Dr. Michael Flythe, and David Gonzalez, and also Dr. Bina Jo from University of Toledo, and our funding from NIH and Crohn's and Colitis Foundation. Thank you.
Thank you, Dr. Kumar, for sharing your studies on the TLR5 knockout mice, and we look forward to continued work and answers using this model. I had one kind of maybe just tangential question. I was interested in the inulin-fed mutant mice and whether or not they were hyperphagic or what their food intake was compared to the non-inulin-fed.
Yes, they are irrespective of whether we give lab chow or inulin diet, they are hyperphagic. Yeah, we did not do calorie restriction in the liver cancer model because it is a 6 months time point. It is very challenging to do pair feeding for 6 months, but definitely by calorie restriction, they can be it can be prevented. Metabolic syndrome can be prevented in these mice. Irrespective of diet, they are hyperphagic.
Yeah, interesting. It makes you wonder what the role of TLR5 is in the signal, right, for eating. Another tangential question, anyway. I think we can move to the panel, and we'll get the rest of the questions for Dr. Kumar and the others. Thank you. And I think Phil will also be joining us. And we'll wait a few moments for the others to join. We want to let you know that the webinar will be archived, and many of the questions and answers will also be posted in the near future after our discussion today. And it may take a lag time for some of these questions to come in. As I've been involved in some of these, that does happen. I have a question for Christian.
Yes.
I'm curious about passive microbiota transfer in humans.
Passive transfer?
Yeah, yeah.
Well, that's
Maybe
interesting.
that's for everyone too, right?
Well, the oral, I guess we know there is, you know, oral and rectal transfer of different diseases. So there's one thing. Yeah, I don't think, I mean, obesity is not, it's not a communicable disease per se. I mean, it's kind of fascinating that you could transfer the phenotype from obese patient to a mouse, right? So, or at least increase their calorie intake for the same amount, or energy producing from the same amount of food. So there is a microbiome component to the obesity that is transferable to experimental model. But you know, is it for a relative, for example? Is it communicable through you know, fecal oral route? The answer will be no to me. But the IBD field has the same question. They were looking at identical twins, one that have no disease and one that has the phenotype. What's the environment role? What's the role of microbes? And I think there were no definitive answer to that. You know, could you acquire through through contact with microbes, for example, or other environmental exposure? And it wasn't clear to me that that was a possible road for disease communication. So, I mean, we do oral transfer in mice because we could do it. For human, that's another ballgame. If you want to do FMT studies of prevention of obesity, yes, I mean, that's part of an experiment, right? In the community, I doubt that something like that happened.
Yeah, we had a similar question during Liza's talk, maybe a little different. Can the gut microbiome of an obese individual affect the microbiota of others in their community, family, roommates who are not obese and eat healthy diets? This may relate to, you know, some studies looking at people who interact together, you know, may also have similar phenotypes.
Right. I mean, it's not an infectious disease, right? So which microbes has to be transferred, and how you know, aggressive the transfer and constant the transfer will have to be in order to observe a phenotype. There is this case report of FMT for C. diff, you know, cure. That's an acceptable way to treat recurrent C. difficile patient through an FMT, and that person received microbes from a relative that had a higher BMI, and it was said that that patient that received that microbiome also developed, you know, a higher BMI and associated obesity pathology, I think. But this is a case report. I think no one has ever observed. FMT has been very, very safe across for a year and a half of observation in the C. difficile field of treatment. So I think obviously they screen these patients so they don't, you know, the FMT that has been used for treatment has been going through a stringent infectious disease, any metabolic problem, any health-related issue. So you get the best of the best in that FMT. nowadays, but before it was relative and we were not making that much consideration. But I doubt that in the community you could acquire a phenotype of obesity-associated pathology just by transfer. Long story short.
Thanks. That's great. We have a question for Vijay. By limiting fat intake, will that decrease bile salt in your model system, I presume? Will that decrease bile salts in the intestine, thereby decreasing cancer risk? Do you have comments about?
Can you repeat the question, Sharon?
Sure. By limiting fat intake, will that decrease bile salts in the intestine, decreasing cancer risk?
It is a good question. Actually, the diet what we use is not high-fat diet. It is a regular 10% diet. We didn't go below 10% fat content in the diet. When we give high-fat diet, it aggravated, but we didn't do any low-fat diet.
Thanks for that clarification. We have another question for Liza. Dr. Makowski, have you yet been able to examine the ICB microbial signature in humans?
Hi, no, we have not been able to do that. I mean, I think that would be fantastic in the same way that you could look at a gene signature, you know, in different subtypes with survival. We have not been able to do that. One thing that I found in the field, as I come from kind of a different angle and I'm new to the microbial field, is that there's so many different papers and there's contradictory information. And even in our own study, when we looked at jejunum compared to cecum, there are different responses. And I think we have to be a little bit more careful in our comparisons and really clear when we're publishing in how we're treating our mice, the diets, and where we're isolating microbes from. And there just needs to be more work for consistent, so we could summarize things in a clear, consistent way across different models and then compare that to patients.
If I may add, because it's a very important questions, and actually Lisa is right. I mean, there's not that much clarity into who is driving a responder signal. If you want to benchmark that, you know, who is driving responsiveness or non-responsiveness, benchmark that to the clinic as a tool for screening patient, the answer is a flat no. But there is data out there. We did a couple of machine learning analysis of published data for PD-1 treatment in human patient, you know, downloaded all the data, look at signals, and there were no clear predictability in the phylogeny. If you use phylogeny as a signal, entry signal to the machine learning, no predictability. In the function, gene function, a little bit, but not enough to say, oh, I could screen now. It's not really. So I think we need bigger cohort. The geography of this cohort is you know, not homogeneous. So you have cohorts in Europe, Asia, different part of US. So, and they are relatively small, 100, 150. So it's been difficult to get a clarity on that. But you could have, there's always a signal coming all the time, you know, Faecalibacterium, Organococcus, Akkermansia. So those are the suspect, and they are used for treatment, but not for predictability. So there's more work to do for sure.
Yeah.
I have another question for you, Christian, before we move on to some questions for I think everyone might be able to provide some insight. So for Christian, what is the impact of a high-fat diet on large bowel transit time, and hence the ability of colonic bacteria to form particular metabolites? And how does this change when moving the obese patient to a more helpful diet?
Wow.
I know that.
I don't know the answer to the transit. I have no idea. That's an interesting, I mean, I will not even know for a mouse. So even less for a patient. But I think, I mean, if the transit is increased, and we know that the microbiome, if you start having a very fast transit and you look at microbiome, will be very different, because these communities don't have time to set up a, you know, some sort of a of a shop, so they always in constant motion. And it's been known that they have, if you do a microbiome analysis on specimen that have a very high transit loose stools, you will have a very different population than if the person has a slower transit. Now, how does it link to a high-fat diet and different movement? I don't know. I have no idea. Maybe the other panelists will answer that.
Yeah, if anyone has any thoughts on that, that would be great. It sounds like a needed area perhaps, or something that could change, response.
And so I have a comment on that. Like if a person takes lots of high-fat diet, so if it is not absorbed, so they may have some kind of steatoria, like a easy bowel movement that may prevent constipation. But it all depends on how much he can absorb. So it is a very difficult question to address, but this is what going to happen, because I did some human studies with high fat. They were drinking some fat. So basically they cannot absorb anything. It results in steatoria, increased bowel movement.
Here's one for everyone. See who would like to begin. How to delineate whether gut microbiome changes cause changes in host inflammatory status, or host inflammatory status causes gut microbiome changes. So this is the cause and effect question, I suspect. So it's
chicken and egg, famous chicken and egg.
Inflammatory, good question. Does anyone have thoughts about how to approach experimentally?
So can I take, Chris?
Please.
Yeah,
Yes,
sure.
thank you, Vijay.
So I think that inflammation can cause changes in microbiota, or microbiota dysbiosis can cause inflammation. So microbiota dysbiosis is a kind of a catch-all term. So even there is small alterations in taxonomical changes can be regarded as a microbiota dysbiosis, right? So I think both are possible. At least in our study, we showed that just microbiota is sufficient to cause metabolic syndrome by inducing low-grade inflammation. So both are possible according to my opinion.
Yeah, I mean, that's correct. I go back to IBD, inflammatory bowel disease, as a flagship of studies in microbiome and host response. If experiment have been done by different group where they take feces from, remission or patient in remission or patient with active inflammation. You put that in a model, then you could see and read some inflammatory response to the host. So the host was fine up until it met these microbes, and then the host react in a way that kind of phenocopy where the microbe was coming from, which means inflammation or no inflammation, and vice versa. If you interact, I mean, what I was showing here is that if you target inflammation with a molecule, then the microbes change. So they are like, it's difficult who was the first hit in the equation, but it's clearly a bilateral component. One could do, microbes could induce inflammation could change the microbial composition. So you could see that as an amplification loop, right? And somehow, if you disrupt that loop, you may gain homeostasis eventually.
But yeah, if they're asking how to do some of these studies, one might think that you could have antibiotic treatments, or like we talked about and showed, fecal microbial transplants.
Yeah,
And then
sure.
if you want to look at host changes, you could either do some sort of inducible knockdown or knockout before or after these antibiotic treatments, or
Right.
do a pull down of an immune cell, like if you're going to pull down a CD8 T cell or something. And then like Dr. Jobin showed, anti-TNF, or there's other drugs out there for like IL-1.
IL-1, NSAID, people have used NSAID. So, I mean, the model is relatively straightforward. The intervention may be more difficult. If you use an antibiotic, is it a wide spectrum cocktail or a specific some sort of selectivity to the antibiotic or niche, microbial niche you want to target versus a specific takedown of a species or a family, for example, and see what happened to the inflammation. So there's different tools, phages, depletion of specific class of microbes, or targeting with an immune suppressive molecule, or like Lisa said, you could read different inflammatory metric on the host or deplete different organism in the ecosystem.
Well, thanks. Great possibilities there. Here's a question for everyone. Can you talk more about practical applications or potential future applications of the information and research discussed today? Who should go first? Who feels like they have something right off?
Where is our chair?
Okay, I will go first. So at least from our mouse studies, what we think is people with dysbiosis, they should not take a lot of fermentable fiber foods. Of course, it is very well known, IBD patients, they have to stay away from any fermentable fiber foods like FODMAPs. That can aggravate IBD flares, or they suffer from abdominal cramps or flatulence and all. So in our case, like in the liver cancer also, we have the now mechanism, now it is under manuscript is under review now. We know which fraction of the population they should stay away from the highly fermentable fiber. So again, why I'm telling processed, highly purified fibers, because it doesn't work like that. You have to eat whole foods. I will give you an example. For example, if you eat a whole chicory or Jerusalem artichoke, which are a rich source for inulin, they do come with a natural inhibitor for bacterial fermentation. Like for example, chicory root has 2,3-difructose anhydride, which limits the fermentation. So, but what the food companies they are doing is they isolate inulin in the pure form, and they supplement and they put it on the fibers and French fries and everything, and they claim it as it is very healthy, but it is not the case. So fibers cannot be isolated in the pure form and mixed into processed foods. So that is our immediate applicability. So maybe in another 1 or 2 years our paper will be out, which will indicate which fraction of the population should not eat fermentable fiber, or at least purified fiber.
Sure, I could mention something that.
Right.
So if it is hard to understand exact roles of the microbiome, but for example, what it appears is that we know obesity can induce immunosuppression, but we don't want to recommend people being obese, when they have cancer therapy. But can we kind of mimic or induce some transient signaling pathway that we're getting clues from the microbiome and clues for these metabolites or whatever crosstalk is leading to this immunosuppression? Could we then just use that as when they're getting chemotherapy or when they're going in for immunotherapy as a transient therapy? And so those might be things that, we're far away from that, but those might be things that could be practical applications that we could learn from looking at our mouse models, comparing that to patient models.
Great.
Yeah, I'll just jump in along the same lines in terms of cancer risk. So it's clear obesity is driving the risk of many cancers, and estimates on the order of 20-25% of cancers you can attribute to obesity. So that's it's a major factor. We've got roughly 43% of our adult population is now in the obese category. Not all of those are going to be metabolically dysregulated, but a good chunk. And so this is a major problem, and it's obesity has become probably arguably the most important sort of preventable risk factor for many of these cancers. So we're in it's a major contributor, and it's very difficult to lose weight, right? That's a challenging thing. So I think, as Liza said, we're not there yet, but if microbiota can be target that's easily manipulated, and we can figure out how to best do that, then maybe we don't have to lose weight to reverse this obesity problem that we're in, and in terms of cancer. And so I think that's the promise here, is that it looks like of all the pathways leading to cancer, it seems like the microbiota related ones may be as responsive to diet as anything we study. And so I think the promise is that we can come up with a way to healthfully change the microbiome in ways that will prevent, kind of disconnect this obesity cancer connection, and hopefully a combination of not only the number of calories we consume, but maybe the types of food that we consume to better, more healthfully shift that microbiota is going to get us out of some of these issues that we're facing with obesity and cancer.
Steve, on that note, how might exercise-induced differences between obese and non-obese microbiomes play a role in addition to diet?
That's a great question, Sharon. We usually don't just eat in isolation of any other thing we do, and I've been kind of complaining that the nutrition people don't factor in exercise sufficiently, and the exercise people don't factor in diet sufficiently. We really need to get a little more together on that, because that is clearly that combination of healthful changes in physical activity and diet are the way to go.
Right.
We tend to isolate them, and we need to stop that and get that together.
For sure, a safe take-home message is that we can't ignore the microbiome when we think about different disease. In the cancer space, 15 years ago, it was a lonely place for microbiome type of study. But now, if you do clinical trial, you will include that component into your analysis and try to gain an understanding of what information you could extract from that microbiome. But it is part of the equation now, and it's still a emerging field. If you think about clinical impact, we have all these correlation, great animal preclinical model, a little bit of clarity and mechanism, but we haven't done any significant clinical breakthrough with the microbiome. Look at all these companies now. They're still waiting for the big shot, but the field has to take microbiome. I'm thinking obesity, but across pathology, take the microbiome as a metric in your analysis, intervention, or whatever you want to study, and put it into the multi-dimension pictures, so that we're going to punch through that enigma somehow.
We have many questions, and we won't get to them. I see we have 5 minutes remaining. We hope to post even some of the unanswered questions, answers to the unanswered, to the questions that weren't answered in the future. So stay tuned. But I wanted to ask the panel if they had questions for each other, or any final comments before we close.
Many. I'll start with one real quick one. Christian, you mentioned you're looking at microbiome, transcriptome, metabolome. Liza, you had mentioned that there's multi-compartments at play here, and so there's crosstalk between adipose tissue and immune cells and the microbiome. So how do we get better at integrating these multi-compartment, multi-level types of regulatory factors? Because it seems to me we've got to get better at that to start to accelerate the pace, particularly in translational research. So any thoughts on that?
Yeah, I mean, for sure, the 16S has been so cheap now that it's just the baseline. You do it because you could do it, but you don't gain that much clarity. So whole genome, metagenome is getting cheaper. So you put that in the integration, but yet it's static pathways. You don't know if they are functional. Then you said, well, I'm going to have RNA-seq on microbes. I will know which genes. So now you're stuck with a big data set that you try to make sense of it through integration of multi-dimensional data. So it's not always giving you a clearer understanding. But I think, and the metabolite to add to the function. So the more we add on, sometimes I think the less it becomes clear. I don't want to be pessimistic, but I mean, we're guilty of that. We do it because we hope to gain a little bit more understanding, but it was not always a bright, shiny light into the questions. You know, we not always gain clarity. The RNA-seq is great to have identified potential genes induction, but what are we going to do with these genes? Could we intervene on these genes? Could we manipulate microbial genes at this point? Or are we going after the small molecule that are produced by these microbes that are associated with a phenotype that is deleterious? We're not quite there. Most companies right now, they are working on microbes that they could identify as a supplement, and they are not targeting a microbial genes. They're not targeting a microbial metabolite. They want to replace an ecosystem or do cocktail designer. So at one point, we'll have to decide, are we getting bigger data to understand the question in a better clarity, or, you know, companies don't want, they don't care about mechanism. They just go, let's see if it works, and then we'll figure it out later. So I do multidimensional scaling, but I'm not, it's not always been to the best advantage. Vijay has been very great success with
Liza
that.
has a.
Thank you.
Then we have to close, I believe.
Yeah.
Unfortunately. We can't.
Sorry. It falls to us when we're publishing that we have to be clear with the mouse models and ages and sex, and then where we're getting the microbes from. And then when we're isolating maybe from tissue, we need to be clear about the controls we're using, and increasingly I think as we delve more into the details, then we can start comparing and collating our data. And that's kind of on the burden of the journal as well as the investigators.
Yeah, I agree with Dr. Makowski. It is already in practice, at least in the basic science, in the mouse research. Even we had to control the bedding materials, the nestlates and the water, whether acidified water or regular water. So everything has to be clearly mentioned in the methods. Certainly there will be some comparison between the two studies. Otherwise, it will be very difficult to compare.
Yeah, there's actually an international effort on that. It's the Aspire guidelines, and most journals are now picking that up. But it's worth all of us taking a look at that from time to time and instilling that in our students to have that front and center. It's a great point.
Thank you. Unfortunately, we must close. Your presentations were very insightful. There's a lot more work to do, and we look forward to hearing from all of you again. And stay tuned for additional posts for the archive webinar, as well as some of the Q&As, all too, for the great questions. We will look forward to seeing you in the future.
Thank you.
Also, and I'll just wrap up real quickly too, and thank you to all our speakers today. Really great talks. I think the Q&A session will be available to everyone, and certainly the obesity working group at the NCI will take a close look at not only the data that you presented, but at the questions that have come out of this. It's great food for thought. A couple of things were posted in the chat during this, including, and I have to promote a new NCI funding opportunity on the effects of gastric bypass surgery on cancer risk. So that link is in the chat as well, or it's PAR 21133 and 132, I think. Again, thank you to all the speakers, and our next webinar scheduled is in December, and you'll be receiving information on that as well. But it was a real pleasure to have this webinar, and thank you all for attending.
Thank you, Phil. Thank you. Thank you very much.
Thank you all.